Thank you everyone for joining us today. This seminar is part of our open source Neuro series, which is in turn hosted in the Worldwide Science Initiative. The series is all about open tools and methods in neuroscience that are on our website, open-neuroscience.com. You can upload projects to that website. You can also sign up to give a talk such as this one about your own open source projects in the neuroscience by completing a short form. We'll welcome your contributions, and of course, if you'd like to follow us on Twitter and share our content, that'd be great. Today we have the pleasure of hosting Ben Morris and Greg Pask from Middlebury College, a small college in Vermont, USA. Ben will be graduating in May with a degree in molecular biology and biochemistry and a minor in computer science. He will then be moving to Cambridge to pursue a one-year master's in pathology, for which he has been awarded a PiSpy scholarship, after which he eventually hopes to pursue a PhD in molecular biology or a related field. Dr. Pask is a neurobiologist interested in the mechanisms and behaviors related to insect olfaction. His lab at Middlebury currently focuses on the evolution of chemical communication in ants and fireflies, specifically the specific genes involved in detecting these social cues. They will now tell us about PiSpy, and you are all welcome to ask questions in the YouTube live chat or directly on Zoom. Speakers, thank you for being here, and please go ahead.
Thank you so much for having us here. It's great to share with an open source community, an open source tool. So we're here to talk about PiSpy, this really new device that we've been working on for a while that we think is a really helpful tool to be able to observe in an automated fashion a lot of the cool organismal behaviors and just growth and development that can occur. And then I'll start, and then Ben will be able to talk about the work that he's done, the considerable work that he's done to get this going. So yeah, so every good story needs an origin story. Every good, this really cool device that we think will help people needs to have some sort of origin story. And for us, that started with some of our favorite animals in the lab, these little Indian jumping ants, Harpagnathos saltator. They are really interesting. They have all sorts of fascinating behaviors, and we wanted to observe a lot of them as they relate to our interests in insect neurobiology. So one behavior that they do is this antennal dueling. These ants are a really cool species because they are considered immortal in that most ant colonies, when the queen dies, the other workers might lay some unfertilized eggs that become males, and then they leave the nest and can form other colonies. But this species, when the queen starts to die or is dying or being less reproductive, will start to have this big dominance tournament to see which worker will transition to this reproductive state. So one of these cool behaviors that we like to see is this antennal dueling. Here you can see it in this video where it's not hurting the other ants, but they're kind of battering them with the antenna, and they kind of get dominance points by engaging in that behavior. Other times, ants that might not be engaged in the duel can bite, just like one we saw right there. And even when it gets away here, it bites it and grabs it again. And they just kind of, if you get bitten, you do not have a high success rate in making your way to being a reproductive. Also, some of these other cool behaviors we see are worker policing, in which an ant that is maybe thinking about ramping up its reproductive status, other workers will grab it and just carry it around for sometimes up to 24 hours. So this ant is getting policed, and it kind of just accepts it, and it just gets walked around the whole nest box here. And it's, eventually it's kind of going to have all these physiological changes as a result of this policing, that it will not be reproductive anymore. So one of the other things that we were really interested in, all right, say it does become reproductive. I'll stop this one here. How do we know that? It starts laying an egg. So this female here, she is protruding this white egg out of her ovipositor here, and in this nice power squat position, we'll be able to see those kinds of behaviors too. The only problem is that these ants are very visual. So when I walk in to the lab and I look at them, you can see they start to track me. They're like little dogs in this way. It looks like they're wagging their tail and they're happy to see me. It's more of an aggression response. But we affect the behavior. Some ants will kind of just go on doing their tasks, but other ants are very distracted and attentive to our presence. And maybe some behaviors we want to look at are happening at night. This is what it looks like being in the ant chamber at night, at all hours of the evening. And if we start to flip on lights and be able to visualize them, it's not only a waste of my time, because I have other things that I like to do at all hours of the day and night, namely at this example, sleep. But there might be a circadian shift that we're introducing if we start to have all these video filming sessions and trying to capture these behaviors. So what we started to do, and this project all started when I was at Bucknell University. This is Marcy Kittredge. She really launched this project forward when we started playing with Raspberry Pis, these really cool Linux-based computers that I've used in my personal life for all sorts of things, for like home video game systems and jukeboxes for my kids, all sorts of fun things. But I started really playing with them in the lab. And here we have some video or an image of Marcy using the Raspberry Pi for the very first day. They've been used across lots of different open source hardware platforms. And we were really inspired by that. What else? We had our first lab selfie. We started to get into this imaging. So this is in June 2018. We got this first lab selfie of all the students in the lab. And this was the start. This is the start of us observing cool and maybe kind of more hammy behaviors of organisms in ourselves. So we really got started thinking about using Raspberry Pis for different automated things in the lab from this paper that Andre and colleagues put out, the FlyPi, trying to create a really accessible and useful and inexpensive tool to use for microscopy, optogenetics, and all these other options. And we really got our Raspberry Pis in the lab with the intention of using the FlyPi to view all these ant behaviors. There's a lot of different components here. And many of these, because it was geared towards fluorescent microscopy and optogenetics and temperature control, we didn't really want that. We had all the structures built into that. And to get this system working, we would have to use all this additional material that we didn't necessarily need. So it would ramp up our costs for things that we couldn't reap the benefits of later. Very similar to commercial hardware and software, where you're paying for a lot of features that maybe you don't need ever, or not in the near future. So we kind of got to thinking, like, could we actually make something like this? And we went back and forth for a long time. And we started thinking, like, well, maybe we could do this, maybe not. We finally, we pulled the trigger, we went for it. And we were very fortunate that we had a College of Engineering on on campus with us. So we walked over to the College of Engineering, talked to Tom Toole here, who really was a wizard in terms of 3D printing and laser cutting for fabrication purposes. And we came up with this first prototype to look at these ants in these tiny little nest boxes. And this was our first PiSpy prototype. I think we had this before we even had a name yet picked out. And what we had here is the Raspberry Pi computer, this little ribbon leading down to the camera, and we were visualizing the ants from the top. We also needed to illuminate them. So we talked with individuals in the electrical and computer engineering department. They helped us fabricate a piece LED PCB here, a printed circuit board that we could use to illuminate with white light during the daytime settings, and also red light. Many of your insects do not see in the red, so we can image in the red while the ants think that it's dark. So we had the different LED light settings here, and more importantly, we had it programmed through the Pi. Our intention was to program that through the Raspberry Pi computer, so we wouldn't have to come in and switch night and daytime settings all the time. So once we had this, we started to scale up and see if we could actually get something that would work. B. Casey and Joanna Ralph Collado helped a lot with this. And we started scaling up, and we had a whole fleet of PiSpys here. We had 16 going at once. We were controlling them all through a single monitor, keyboard, and mouse through a KVM switch. And it was a lot of wires, a lot of mess, but it was working for us. And we were able to kind of monitor all these colonies, these ant colonies in parallel. We started to share this with the community, presented at conferences, and a lot of people really were interested in this idea and had use for this very simple idea of just, let's just take videos and images. And we were mostly taking videos at this point, but we thought, hey, could we actually go full, not just taking what we had seen demonstrated with the fly pie and doing it for ourselves and kind of keeping it internal, but if there is this much use out there, could we make a nice contribution to the open labware movement? So this idea of, can we take PiSpy from our lab to any lab? And I think this is a common theme throughout all sorts of open source hardware and software that's out there. And that needed to incorporate a lot of different things. We talked to different people about different things they might use this device for, whether it was watching plants grow, tracking feeding of different animals, tracking mating. This is an interest of a close collaborator of ours in terms of firefly mating, and how frequently that occurs and what triggers it. So we started to kind of assemble the goals that we wanted for an open source platform: affordable, flexible, and accessible. For the affordability, we want to make sure we're using inexpensive materials. 3D printing filament, it can be very cheap, especially if you have access to a 3D printer at your institution. And also laser cut wood. Wood is, even though wood is slightly increasing in cost in the pandemic, it's still much cheaper than a lot of other materials. And with that, we wanted to have this low cost option for a single device that could enable a lot of different things in education. Labs that might not have a lot of funds that they can pour into this. Also, if it's lower cost, maybe you do have the means, instead of paying a lot of money, like $10,000, $20,000 for a single device, you might be able to use this option and get multiple devices, do a lot of this work in parallel, and save a lot of money and time that way. Also, the flexibility, the scalability. We wanted to go down from the simple use of, hey, we just want to take automated imaging of our behavior or organism of interest, where it could also scale up to the complex nature of, oh, we want this only to trigger when it triggers it trips a motion sensor or an infrared brake beam or something like that. So some sort of input trigger. And also, we could control the lighting through it. Maybe you have external lighting control and you don't need that. You can still use this device for that purpose. And then all the input and output connectivity really enables a lot of the complexity here. And Ben will be diagramming a little bit of that as well. And then also making it accessible. Having that graphical user interface, the GUI, to be very easy to use. I myself have zero Python coding experience. This is where Ben really carried us. And I can go in and use this, no problem. People have already started to use a device like this, and because of its accessibility. So that was one of our major goals here. And then to have those who are experienced in coding, to have it be a sandbox. They can play around, they can access and fork the repository, start tinkering around, and then sharing that alike, and really starting to develop this nice community. So we wanted to have that kind of ingrained in this design. And we ended up with a modified design here, still based with a lot of 3D printed and laser cut components. But we have this is our current model, our non-specialized model, which you have the Raspberry Pi mounted on the back. There's a little thumb screw here that you can raise or lower it however you want. And it's supporting it. We have access for all the ports that you'd need for a Raspberry Pi 3 or 4. There's also very simple modifications you could have for a smaller Pi, like a Pi 0. The camera is mounted. We just wanted to look at the ants from the top down, but we have it on a ball and socket joint. So you can pivot that camera to point at any angle and maybe set up multiple devices looking at the same specimen at different angles, maybe a top down and a side view. Have that flexibility there. And we also integrated that white and red LED into it. That was, what we built. There's also very commercial, accessible, easy, and inexpensive passive infrared things that Ben will talk about later. That's also depicted here. But what we really needed, and all of this came down to about $100 US. So we like that price point, and it's really we're using the bare bones here to make it just what you would need to observe these organismal behaviors. Now, to control all this, we needed this graphical user interface. And I think when I first met Ben, I shared this, little idealized window. This is not an actual graphical user interface. This is just what I wanted. This is my thoughts and feelings of, hey, this would be cool if we had these kinds of buttons and text boxes to input our settings and whatnot. And we were at something that could work. It was kind of clunky. And that's where we were at. I moved, transitioned to Middlebury College, and this was during COVID. And this project almost died then because we had no real reliable graphical user interface. And then I was approached by Ben, who was doing a computer science minor in addition to his biology interests. And it was a perfect match. And I'll let Ben take it away here. Though we do have maybe some questions here. Oh, these are links. Oh, okay, perfect. So Ben, take it away.
Yeah, so I came onto the PiSpy project, as Greg said, and kind of the big remaining body of work on this project was to develop the GUI that Greg just showed in an actual functional form. So we did all of this using tkinter, which is a Python GUI library. And this is what we came up with. So obviously a little more complex than the simplified diagram that Greg initially showed me, this one here. But we tried to keep it as streamlined as possible, just allowing you to control the basic settings of the PiSpy for a variety of different uses, which I'm going to get into now with the rest of this presentation. So the most basic way you can use the PiSpy is just instantly recording, basically the equivalent of taking a picture with your camera. This can be really effective for testing settings for the PiSpy if you're going to then use it for longer periods of recording, or if you have specific behaviors that you want to capture at specific times. So for instance, we put two crayfish in a novel tank and they hadn't kind of been introduced to each other yet, and then can record this aggressive behavior as they were kind of getting acquainted and establishing dominance. Or alternatively, motion tracking is a really powerful tool. So we put 10 Drosophila larvae in an agar plate with apple cider vinegar on this filter paper in the middle, and then used image tray to track their motion over the course of 10 minutes. And that's manual image tracking, which is really easy to use. I'd never used image tray or any kind of image processing software. You basically just point and click and then go frame by frame. So that's pretty powerful and pretty accessible. If people have more specific uses, there's a lot of free or open access software like Deep Lab Cut that's available. This uses machine learning to track, say, individual body parts within an animal. It's a lot more powerful. And yeah, it could be used with the PiSpy, and the images generated by the PiSpy or videos generated by the PiSpy to do a lot of tracking and kind of other behaviors like that. Deep Lab Cut's really cool because, again, you can track the motion of, say, specific limbs of an animal, or in this case, they're looking at different parts of the mouse. So you can really quantify certain behaviors. Another awesome way to use the PiSpy, and something that we've had a lot of fun with, and also kind of in our sense start to generate sample almost experiments, is through automated imaging at set time intervals. So putting into the GUI, again, we're trying to make it as streamlined as possible. So you say you want images over the course of 300 hours, take an image every 15 minutes. And then you just run that. What you can get out of that are really fun time lapses where you can monitor the growth of an organism over time. So here we have bacteria that we isolated from the soil. We got some help from the microbiology professor at Middlebury with experiments from her class, and then used that, poked 4 colonies onto an agar plate and watched it grow. And again, we're not doing any quantitative experiment here because we're just trying to demonstrate the utility of the PiSpy, but it would be really easy to imagine how you could, say, quantify growth over time by looking at the area of the plate that each colony is taking up. And if you had different growth conditions or different types of bacteria, you could do comparisons that way. On the right, we did a very similar experiment, but growing beans in these clear plastic planters. So we made these planters with the help of Eamon McMahon, who's the scientific machinist at Middlebury, but you could easily do it in commercially available planters as well. But what this really was fun for us and allowed us to do was visualize both the root growth under the soil, because we could put the seeds right up on the edge of the planter, and then also see the plants actually growing. We're not plant biologists, so our plants didn't always grow, and we probably could have had slightly better growth conditions, but we got some fun videos out of it. And I think that just, trying to think of different ways PiSpy could be used in more formal experiments. You can also, as Greg talked about some earlier, use external sensors to trigger recording or imaging. And again, that's really simple to use or to implement with this GUI, just specifying, in this case, video settings. Raspberry Pis, and I'll get into this, I think, on the next slide. Raspberry Pis have a number of GPIO pins, which are general purpose input and output pins. So you just plug the sensor into one of those and can specify the delay, so you're not, if a motion sensor is triggered, you don't immediately re-trigger it. So here's how you'd plug in a motion sensor. Again, they're really cheaply available on sites like Adafruit. And in our user's guide, we tried to clearly diagram how to plug them into these GPIO pins, both with this kind of pinout diagram and then the actual Raspberry Pi. And most sensors, like a motion sensor, the next example we have is an IR brake beam, but there's lots of other ones as well, like pH monitors or temperature sensors or force plates. There's a lot of different ways you could go with that, depending on the specific settings or the specific experiment you want to run. And they're, again, really easy to plug in and can be used for a lot of applications. We only went as far as kind of setting up a motion sensor to see who was walking around in lab, just kind of fun experiments like that, and didn't do anything more formal with it. But there's so many applications for this kind of motion sensing and other sensor-based imaging and video recording. The other one we used a little more in our testing was IR brake beams, which work with, they have an emitter, which is emitting an IR signal, and then a receiver, which receives the signal. And those both plug into the Pi. And again, there's just the one input pin that, or input wire that is actually telling the Pi when the brake beam is triggered. Again, these are really affordable and easy to get on a site like Adafruit or Amazon, or really wherever you can find kind of scientific supplies. And for us, we use that to study feeding in ants. So we modified our standard nest boxes. So we had the nest box here on the left, where the main colony was. And then on the right was a smaller chamber that we put crickets in when we fed the ants. So it's like a foraging arena. And connecting them was this little tube that ran past the brake beam. So the brake beam is housed. This was a really fun project for me because I got to play with Legos, which is something I loved to do in middle school and hadn't done in a long time. So that was a fun couple of days in lab, trying to set up a kind of a holder for the brake beam to go through, or just to hold the brake beam with this tube running through it that the ants could walk on. And the way this worked was when we fed the ants, they'd walk over to the foraging arena and trigger the brake beam. So we'd see them walking there without food, and then they'd sting crickets and carry them back to the main colony, carrying the crickets. So again, we weren't generating quantitative data in this case, more trying to demonstrate uses, but would make it really easy to, for instance, measure how many times the ants are going to get food, or how many ants go into the foraging arena versus staying in the main colony, or kind of other studies of ant food and foraging, or ant eating and foraging and hunting dynamics. So here's what we actually came up with in terms of the video. You'll see this little index card, kind of what seems to be already breaking up the brake beam. That was, in our testing, the brake beam wasn't quite sensitive enough to be triggered by the ants, but putting a little bit of paper, or in this case, an index card, between the emitter and the receiver blocked just enough of the IR light that the ants would trigger the sensor. But when there wasn't an ant there, the sensor wouldn't be activated. So that was kind of just fun little tinkering to make this setup actually work for our purposes. And you can see when the video plays that we now, so it started right as the ant was crossing the brake beam, and we can see a bunch of ants walking back with crickets, and then this one little cricket trying to make a dash for it from the foraging arena without realizing that all it's doing is running towards a number of, or another ant chamber with even more ants, and probably a not so fun fate for it. So on top of all those kind of uses of the basic PiSpy, what's really exciting about this project to us is how customizable and flexible it is. And that honestly starts for us with what was originally the first design of the PiSpy. So in our case, we really are using these devices to study the Indian jumping ant. So we don't need some of the functionality that came from the more general model that we now have as like our base model. So we have this, it's only ever imaging from the top with the LED PCBs attached, because again, that white and red light cycling will mimic day-night cycling for ants. And we have the containers, or the stand nicely fits around the nest boxes that we use, because we're always using nest boxes of the same size. And so in our lab now, we've downscaled a little from the 16 PiSpys that Greg had going at Bucknell. We're now down to 4, with 4 different colonies being imaged. But again, using the KVM switch is a really easy way to have multiple colonies, multiple PiSpys going at the same time with only having to use one monitor. So the night cycling, again, it comes out of that specific use for us of looking at organisms that can't see red light. So we have these PCBs that, again, by following the wiring diagrams in our user's guide, can be pretty easily plugged into the Raspberry Pi's GPIO pins. And those are controlled then with the GUI to be on and off at set times. So by default, we'll do something like 7 a.m. to 7 p.m. has the white light on, and then 7 p.m. to 7 a.m. the red lights will be on. And again, that's just mimicking light and dark for the ants because they can't see the red light. And that leads us, or allows us, to observe behaviors 24/7. So instead of having to, say, come in every hour to lab to see if the ants are doing a specific behavior, we can just record videos at those times, or at whatever time intervals we want, and then analyze that data later, which is way easier, even if it does result in mountains of videos to look through at the end. So we can observe behaviors like the antennal dueling Greg was talking about earlier in our ant colonies. And at night, we can still see, even if it's a little washed out in the red light, we can still see the limbs of, or the legs of the ants, and the movement of the antenna, and their feeding or sleeping or whatever they're doing at night. We can still visualize that without having to go into lab or disturb the ants. The camera settings that we have programmed in are what was working best for us in our testing. And in the interest of keeping the GUI as clean and simple as possible, we didn't put in kind of ways to modify those settings in the GUI, but it's really easy to do in the code. And again, this is all instructed in the user's guide. And I tried to also, in writing comments for the code, make it clear what different lines are doing. So if you do need different settings for day or night recording, you can go into the actual Python files that we have up on the GitHub and change that relatively easily. Yeah, and I really tried to make that as accessible and simple to do as possible, even for someone who hasn't necessarily had much prior experience in Python. And then PiSpys have also proven very effective in course-based undergraduate research experiences and other academic settings like that. So on the idea of kind of modifying the PiSpy design for specific uses, this is one that was done by Andrew Ng and Anthony Marinello in Greg's entomology class last fall. They were looking at cricket feeding and how circadian rhythms affect that. So they had this setup where it's basically a mount for the PiSpy that goes directly onto the lid of a box or like a Tupperware or something like that. And they could put one on either side of the box, with the lights also mounted underneath. So they could do a condition where it was always white light, a condition where they did the light cycling, condition where they had the white light on one side and the red light on the other side. And then they could image where, or they could image the food sources to see where, which food sources ants were eating or how much they were eating or things like that. And then another project from the same class, from Sophia Marlino and Jen McGann, was looking at the feeding of these hornworms and how they would choose food sources based on the presence of artificial predators or predator mimics. In this case, these little printouts of wasps. They had a color wasp, a black and white wasp, and an actual pinned wasp. And they were looking at how whether the hornworms would choose a nicotine-laced food or the regular food source, depending on the different predator conditions, with the basis being that when these organisms use nicotine as part of a predator defense mechanism. And by the ease of the PiSpy allowed a group like this to have multiple conditions running at the same time. And again, it was all kind of organized through the KVM switch, so they could just do one computer and then analyze all that data after. I also used the PiSpy for one of my classes. I had a biostats and experimental design course last spring, which was when I was kind of ramping up some of the work I was doing with the PiSpy, especially these bean time lapses. And I realized that it was, we had an independent project at the end of the class, and I realized that PiSpy was kind of the perfect opportunity to, or that project was kind of the perfect opportunity to use the PiSpy to generate some quantitative data and kind of test its use in measuring bean growth. So I was looking at the common bean, again, the same organism we were growing earlier, and was able to generate a number of metrics. So I was looking at time to germination, which was just when I saw kind of movement from the seeds, the maximum height that the plants reached, and then different metrics of growth rate, the first being just from 0 to 2 centimeters, so the first part of the bean growing, and then the rest of the growth. And you'll see in all of these plots that I did not really generate any interesting findings. I was comparing control versus salt water for the beans, and I really didn't get very much. And I think that's because I don't really know much about plant biology, and I did not take the best care of my plants. I tried to just water them once at the start to try and minimize watering as an external factor. And the result was that they all just kind of were lacking in water and didn't really grow. So I didn't generate the most interesting data, but PiSpy was very effective in doing exactly what I told it to do, which was take pictures of the beans for me to then analyze. I think I failed more in my care of the beans themselves. Awesome. And then what we're really excited about with the PiSpy is that we think other people can take this base model that we've developed and use that for sure, but also then generate their own modifications. We worked a lot with Andre, who's at Open Neuroscience, to license it in a way with the Open Source Hardware Association. The code is all licensed through this GNU GPLv3 software license, and then the CERN open source hardware license, which is strongly reciprocal for the hardware. So all of that is to say, anyone can use the PiSpy, anyone can modify the PiSpy, and any modifications done to the PiSpy need to then be made open source and open access. So we're really hoping to kind of generate a global network of people using PiSpy, taking our base model and changing it for their own specific uses. And as part of that, we set up a forum on GOSH, Gathering for Open Science Hardware, where people can do Q&A, troubleshoot things. Greg and I will both be checking that pretty regularly. Even going forward after I graduate, I'll stay active on these forums and on the GitHub. So we're hoping people can take this base model that we've developed and turn it into something really specific for their uses, and then share that with the world and with other scientists, and hope that kind of sparks new innovation and new projects. So some things that have already come to mind for us, in our case, we can use the red lighting for studying behavior at night. That's not necessarily an option if you're studying mammals that can see red light, because then you're be totally disrupting their circadian rhythm. Raspberry Pis have an IR camera, they call it the no IR. And we've already actually heard from someone who set up the PiSpy with the no IR camera, and it's working great. So that's an option if you wanted to use IR light sources and that camera. We've also thought about but never implemented the PiSpy as a camera trap. So hooking it up to a battery pack, creating a slightly more weatherproof case, and then you could put the PiSpy out in the field and use the same sensors we were using in the lab, like the brake beams, or especially the PIR motion sensors, which is what most camera traps use. And PiSpy could be slightly modified to become an open source, affordable camera trap, which has a huge range of utility in ecology studies or any kind of wildlife studies like that. So that's all we have today. We've done a lot of fun time lapses as well. So here we have PiSpy spelled out in E. coli. But yeah, really appreciate everyone's time. And if there's any questions, we can do that now. Before we go there, though, it's worth highlighting just how collaborative PiSpy has been as a project. We've gotten a lot of support at Middlebury College from people in the CS department, like Andrea Beccari, the other members of the Pask lab, other people in entomology and the STSS at Bucknell as well. A lot of people Greg highlighted earlier in the talk, and Andre over at Sussex has done a lot of help on the back end with editing and letting us or helping us navigate the open science world and make this truly an open source project.
Thank you. Thank you for that wonderful talk. I think it was amazing. Very, very fun, first of all, like all the different organisms and really the PiSpy is super neat and flexible, as you described. And I think it was a very, very complete talk that will give everyone an overview of everything that can be done. So I wonder if there's any questions from the audience here in the Zoom room or anyone that might be on YouTube watching us live.
I just want to say, I joined late, so I just want to say a quick hello. But the video is breaking up a little bit, but can you guys hear me?
Yes, we can.
Great, I just thought I'd do a quick shout out. I think this is a great, fantastic project. I love the interdisciplinary nature of it. I love the open source nature, the creative ideas, and the way it's come out. And it's great to see Ben and Greg. Just wanted to say a quick hello. And I'm sorry, I joined a little late, so I didn't hear the beginning.
Thank you. You can catch anyone that hasn't been able to watch the whole talk can catch it on YouTube later on. It's going to be there, and you can interact with Ben and Greg also on Twitter and everything. I really liked your repo. I enjoyed all the care that has been taken in making everything being spelled out there. And I think that's truly an example for other projects. I was wondering when you mentioned the different projects that the students did they build it as well, or were they using PiSpy that you had in the lab?
Yeah, I can handle that one. The hornworm one with all the little caterpillars feeding, that was just with what we had existing. There were still some modifications, but that was kind of a, hey, we had them around, and it was even our unit that we used primarily for the ants was, we could just put something on top of that little box part, and that's where the caterpillars were. The cricket feeding, where we had the lid, that was purely made, the hardware, like the additional hardware was made from the student's perspective. And they kind of really gravitated towards that. I'm like, all right, measuring how much clearance do we need for the ribbon? How do we get this so it's air, how we can control the lighting and not have any ambient light. So they went and got them some spray paint, and they sprayed down this whole storage tub and everything. So we had, as college campuses and different institutions are kind of getting more makery and there's different maker spaces, we actually think that this could be a nice thing to, hey, we could develop and make something in this available space on campus that you could then use for anything you want to do in the future, right? Once you have those tools and you can think like that in the engineering fabrication mindset, there's a lot more problems you can find solutions for.
Yeah, definitely so. And I think the educational approach of putting it together and trying to find those solutions is also something that you've made very, very user friendly in your design, and and besides the fact that it's low cost, and yeah, I think it's a great package all combined. One other thing, when you mentioned the adaptations, of course there are tons. But were you thinking maybe that it could be head mounted or like microscopy applications? Have you worked with any other stuff, other people that have made more adaptations than the ones you showed, or where do you think that might be going in the future?
We have, I mean, we put this out last month, so we really have no idea where people are going to take this, and I think that's kind of the exciting aspect of it. Like my family and I think we might be looking at it using a motion sensor to see when our dog is eating and drinking and like that kind of stuff. So having kind of more community science and citizen science angles, I think is a really nice way to apply it. And when it's easy to build and it's not too obtrusive, you can have people design, like, community science projects where you're trying to design the best way to mount this to serve that specific needs. I know there's projects where it's like you put different food out in your backyard and you see what species of ants are coming to and how often they're coming there. This could serve that. You could either, just get a battery, like an extension cord, and run it out and hopefully give it some protection from the elements. But yeah, like we have already had people contact us that were had a lot of the hardware built with another open source Raspberry Pi based project, and they just plugged the software in and they like how that works a little bit better for them. So there's also a lot of a lot of more opportunities for kind of mixing and matching a lot of these open source hardware and software options, and to see what works best for your needs.
Yeah, I think that's super inspiring. And maybe if no one has any other questions, we can close the live streaming with that. Thank you so much. Sorry.
Thank you.
Thanks so much. This is great.
Thanks for coming, Jamie.
Yeah, good to see everybody. Really fun.
Me too.
Hope to see you next time on May the 4th for Professor Suhasa Kondara and Mayan's seminar titled Open Source Neurotechnologies for Imaging Cortex-Wide Neural Activity in Behaving Animals. And that will be again at a regular time of 15 UTC, just half an hour earlier than today. So I'll just close the stream. If you'd like to keep talking, no problem. Thank you so much.
Thanks, Cecilia.